When a muscle cell is depolarized, it's said to be excited, and an excited muscle cell is going to do its job, which is to contract. So action potentials in muscle cells lead to the sliding action of the myofilaments. So remember, the action potential was moving across the sarcolemma or the muscle membrane, which simply means that a wave of depolarization is moving in a localized manner from one area to another. Depolarization is the movement of those positively charged ions from outside the cell to inside the cell. So as that is happening along the sarcolemma, the T-tubules, which are a part of the sarcolemma, are also depolarizing. As they depolarize, they stimulate the sarcoplasmic reticulum to dump out calcium ions. The calcium ions are then going to start to interact with these small little contractile proteins. So let's look down here at what's going on. Here we've got our myosin heads, we've got the actin, the three little blue balls are the troponin, and the little string-looking structure is your tropomyosin. So under normal conditions, when this muscle cell is relaxed, the troponin holds the tropomyosin over those binding sites on actin so that the myosin heads can't attach. But when calcium is present, then the calcium binds the troponin changes shape, and the tropomyosin moves off those binding sites. So as long as calcium is present, the myosin heads can bind to the actin. So that takes us into the sliding filament theory, which is what happens once those myosin heads bind to the actin. Myosin heads include an ATP binding site that functions as an enzyme that breaks down ATP to ADP, adenosine diphosphate, and a phosphate group. That enzyme, when it breaks down ATP, releases energy from that hydrolysis reaction, and that energy stored in the myosin head to be used during the contraction cycle. So the myosin head is said to be energized when it's containing that stored energy. So that stored energy pulls that myosin head back into what we call a cocked position. So it's sort of like stretching a spring. But also notice that we've still got the ADP and the phosphate attached to that head. The second step is where that energized myosin head attaches to the myosin binding site on the actin. So it releases that energy that's holding it cocked back in that spring-like position. The head is then referred to as forming a cross bridge. So the cross bridge is actually what is formed here when the myosin head binds the actin binding site. After the cross bridge forms, the myosin head pivots a little bit, and as it changes into the new position, it pulls the thin actin filament past the thick myosin filament toward the center of the sarcomere. So it's generating this tension or force in the process. This is what is known as the power stroke. So here you can see how the myosin head has changed its position. As it does, it's pulling on that actin filament. All right, so again, that's what's known as the power stroke. And the energy required for this power stroke is what we got from that energy stored in the myosin head when it hydrolyzed or broke down that ATP. Once the power stroke occurs, the ADP is released. All right, so now that head is not bound to ATP or ADP. So at the end of that power stroke, the cross bridge is still firmly attached to the actin until another ATP molecule comes in. So remember, here we took off the ADP, but now we want to detach that head, so it requires another AT molecule coming in. As the ATP molecule comes in and binds to that binding site on the myosin head, it detaches from the cross bridge formation on that actin. So what happens? The whole cycle starts again. The ATP is hydrolyzed to ADP and phosphate. Okay, the head is in the cocked position. Then the cross bridge forms using that power stroke, okay, pulling up on that actin molecule, which then causes that sliding action to occur. The ADP is released. So now the myosin head is free to bind another ATP, which it then hydrolyzes into ADP and phosphate and goes back into the cocked position. Now all this is going to continue to happen as long as two things are going on. There's plenty of ATP to break down, get back into the cocked position, power stroke and bind again, and the cycle continues. But also the important thing is there has to be plenty of calcium available. Remember, this all started because calcium was binding to that troponin and opening up those binding sites. So there has to be sufficient ATP and calcium available for this process to occur. So just a short little summary here to put it into words on a slide, I guess. The sliding filament theory consists of four major phases. The cross-bridge formation, where the tension forms, the myosin head attaches to the actin binding sites. Then there's the power stroke, which pivots and pulls the actin filaments toward the center, causes the ADP and phosphate to be released. The cross-bridge detaches then, so the head of the myosin detaches from the actin as a new ATP arrives, and then the head goes right back into that cocked position, ready for the process to start over as the ATP is then hydrolyzed back into ADP and phosphate. So here we have sort of an overview of all of this on one slide, but it also includes how this action stops. So remember, we had an action impulse coming in from a motor neuron, causing acetylcholine to be released. The acetylcholine bound those receptors on the sarcolemma and allowed for sodium to come in. As sodium came in, it triggered an action potential in the muscle cell, which then caused the calcium to be released from the sarcoplasmic reticulum. The calcium then bound to the troponin on the thin filament, exposed the actin binding sites so that the myosin heads could then bind to the actin. The myosin heads bound to that actin undergo the power stroke and release. The thin filaments are pulled toward the sarcomere. So that's as far as we got on the previous slide. But we did mention the fact that this will stop once the calcium levels fall in the sarcoplasm and once the ATP has been used. So calcium has to be pumped back out of the sarcoplasm and stored back into the sarcoplasmic reticulum. So that requires ATP to do that as well. Anytime you're powering a pump, remember you've got to use energy. So ATP actually has two roles here. One, the ATP is used for the cocking and the releasing of that myosin head, but also we have to have sufficient ATP to power the pumps to move that calcium back into the sarcoplasmic reticulum for storage. Once the calcium is pumped out, the troponin goes back into its normal shape. The tropomyosin then covers the binding sites, and the myosin heads can no longer bind to that actin and cause the sliding filament action to occur. That is when muscle is said to relax.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin,